So we're going to talk now about the second stage of cellular respiration, which is the breakdown of pyruvate. And this is going to be your favorite one because it's the shortest one. And it starts with pyruvate entering the mitochondria. So remember that the pyruvate was made in the cytosol because that's where glycolysis occurs. So it needs to enter the mitochondria. And so this is a transport process. There's a symporter that will move the pyruvate into the mitochondrial matrix. So the breakdown of pyruvate is going to occur in the matrix of the mitochondria. And once the pyruvate gets into the matrix of the mitochondria, in one big chemical reaction, the pyruvate is broken down. So remember, pyruvate is 3 carbons. It's going to be broken down into a 2-carbon acetyl group. And the 3rd carbon is going to be let off as CO2. So here's that CO2 being made. So the 2-carbon acetyl group is going to be attached to a placeholder called coenzyme A. So that's what gives us this acetyl-CoA. Acetyl-CoA, again, is the acetyl group, 2 carbons, stuck onto a placeholder called coenzyme A. At the same time, same reaction, there was a redox reaction where the NAD+ was reduced to NADH and the pyruvate was oxidized to the acetyl group. So remember, this is a reduction, this is an oxidation, that gives us the redox reaction. So just to summarize, it's a one big step reaction that breaks off a carbon, releases CO2, puts the other 2 carbons onto coenzyme A, and reduces NAD+ to NADH. So for each time this reaction occurs, the input is pyruvate, output is acetyl-CoA, CO2 is our waste, and we used an NAD+ to make an NADH. So make sure you're putting that into your table of inputs and outputs. That is per pyruvate. So remember that in order to know what it is per glucose, we have to multiply by 2. So per glucose, we used 2 pyruvates, we made 2 acetyl-CoAs, we made 2 CO2s, and we made 2 NADHs. So from here on out, we're always asking ourselves how many times per reaction and how many times per glucose. The third stage of cellular respiration is a citric acid cycle. So this is where the acetyl-CoA is going to head from the breakdown of pyruvate. So acetyl-CoA is coming from the last stage. The citric acid cycle is a metabolic cycle, and what that means is that something that is used at the beginning has to be remade at the end. So this is a lot like a cycle that you're probably familiar with from elementary school, the water cycle. So remember, like in the water cycle, you have rain come down from the sky from condensation, and then the sun heats it back up and it evaporates and comes back into the air, and then it recondenses to reform rain. And that is the water cycle over and over again. It evaporates, goes up, it condenses, and falls back down as rain. So something that's used is remade at the end. So what's going to happen at the very beginning of the cycle is the acetyl group is going to get attached to oxaloacetate. And oxaloacetate is what is used and remade. Okay. So the acetyl group is 2 carbons. The oxaloacetate is 4 carbons. So this is going to create a 6-carbon molecule. This will then initiate a series of steps that will shed 2 CO2s. So remember, that was your acetyl group. Your acetyl group is being converted into CO2s. It will produce 1 ATP, 3 NADH, and 1 FADH2. This is per turn of the cycle. So remember, per glucose, we have to multiply by 2. So we get 4 CO2s per glucose, 2 ATPs per glucose, 6 NADHs per glucose, and 2 FADH2s. At the end of the cycle, the acetate is remade because, again, this is the cycle. And this is occurring in the matrix of the mitochondria. So we'll just look at the citric acid, and this is as much detail as we're going to look at it, watching what happens to the carbons. So here's the acetyl group. It's 2 carbons. It's on that placeholder coenzyme A, but nothing is going to happen with this coenzyme A. Like I said, it's just a placeholder. So those 2 carbons are going to get attached to the 4 carbons of oxaloacetate. So 2 carbons plus 4 carbons is going to give us a 6-carbon molecule, which is citrate. A series of redox reactions are going to occur. How do I know this is a redox reaction? Here's NADH, here's NADH. At the same time that there's the redox reaction, a CO2 is broken off. So if I take a 6-carbon molecule and break off a CO2, that leaves me with 5 carbons. I then do another redox reaction and also break off a CO2. So now I'm going to have a 4-carbon molecule. I'm going to strip some more energy from that molecule by pulling off a phosphate. So don't worry about this too much. It's an intermediate that can eventually be used to make ATP. I'm then going to have one more redox reaction. So here's FADH2. Remember that FADH2 is created by reducing FAD. So that's how I know it's another redox reaction. And then later on I have another redox reaction, right? So there's another NADH being made. And then all of this has also been just changing the forms of the carbon to get me back to oxaloacetate that I started with. So I had to use this at the beginning. I'm remaking it at the end. I added 2 carbons and I lost 2 carbons. So I added the acetyl group, which had 2 carbons, and I lost 2 CO2s, which is 2 carbons worth. So this extra 4 carbons, that just keeps cycling and cycling and cycling. It keeps getting used and remade, used and remade, used and remade. I always just have 2 carbons coming in and 2 carbons leaving. So, and in this process, and this is kind of the key, coming back to energy, I took all of the energy that was left in glucose and put it into either NADH, ATP, or FADH2. So at the end of the citric acid cycle, remember I started with a 6-carbon glucose at the very beginning of cellular respiration. I lost 2 carbons in the breakdown of pyruvate, and now per glucose, remember I have to multiply by 2, I lost 4 carbons in the citric acid cycle. So 4 and 2 is a total of 6 carbons. So I added 6 carbons from glucose, and now I've lost 6 carbons as CO2. So at the end of the citric acid cycle, there is no more carbons left from glucose. All of the energy from the glucose has been put into energy intermediates, either NADH, ATP, or FADH2. Anything that's been put into ATP is good to go. The cell can now use this. But the NADH and the FADH2 is still not usable for most of the cell's work. So the next stage of cellular respiration is going to have to take the energy from those molecules and turn it into, or transform it into ATP energy. So at the end of the citric acid cycle, where has most of the energy gone? Not to ATP. Remember, most of it is now in NADH. Remember that NADH is storing energy in the form of electrons. So in order to get the energy out of NADH, I'm going to have to carry out more redox reactions where I take the electrons from NADH. And so that opposite reaction, NADH to NAD+, will be the beginning of the next stage, stripping that energy out of it. The second question here, what cellular processes produce the CO2 that we exhale? This is just to remind you that the reason why we have to breathe in oxygen and breathe out CO2 is because of cellular respiration. So at the end of the citric acid cycle, remember we lost 2 CO2s in the breakdown of pyruvate, and we lost 4 CO2s in the citric acid cycle. All of that CO2 is going to have to be breathed out with our bodies, or by our lungs rather. So up until now, have we used oxygen yet? If you think back, hopefully you realize that so far we have not used oxygen. So we have to breathe in oxygen, but the reason why is going to be in the last stage. Stage 4 of cellular respiration, the oxidative phosphorylation phase. And that's what we'll be talking about in the next video.